In this 3 mark subjective second optional question, they are directing us to describe the amplification process of the gene of interest using polymerization chain reaction technique. For this, we are given the detailed amplification process with the illustration of restriction site of the vector DNA and of the digital DNA. In this both this DNA, we can find the restriction site is exactly existing as a inverted repeated sequence or a palindromic sequence. For this narration, we are getting 1 mark. This restriction site is taken to the cleavage by the restriction endonuclease enzyme of E. coli, so that we are getting the sticky end. For this narration, we are getting another 1 mark. In the third step, we are applying the DNA ligase so that this cohesive end of the vector DNA as well as of the residual DNA will go for linkage, so that we are getting the recombinant DNA. In this way, the developed recombinant DNA is taken to the E. coli plasmid, so that the E. coli, when it is multiplying, it will favor the expression of this recombinant DNA, so that we will be getting the digital product. For this narration, we are getting another 1 mark. In this way, we are satisfying the overall 3 mark demand of this subjective question. In this 3 mark subjective question, they are directing us to describe the amplification process of the gene of interest using polymerization chain reaction technique. For this, we are given the CBSE pattern PCR diagram to represent the amplification process. In the first denaturation process, the double-stranded DNA is allowed to get unwinded. For this, we are giving the temperature up to 95 degrees Celsius. In the second step, when we are completing the annealing process, the primer are allowed to attach at the 3' end of the unwinded strand. In the third step, during the extension execution, we are making the new DNA strand synthesis by applying the DNA polymerase enzyme of Thermophilus aquaticus bacteria. In this way, we are repeating this 3 steps to get the double-stranded DNA, which is the exact copy of the given DNA. For this illustration, we are getting 3 mark.